Hello, everyone, and welcome to this webcast. My name is Farup, and I will try to keep you entertained for the next 20 minutes or so. But before I get started on my presentation, I just want to take the opportunity to thank the organizers, the Danish Diabetes Academy, and the Metabolic Research Network at Cambridge for organizing this event even under these difficult circumstances. I'm sure that it's going to be a great meeting, and really look forward to it. So, with no further ado, I will share my screen and get started on my presentation. So, um, the quest for eternal youth or immortality is something that I think humankind has been fascinated um, about for many years, and it's quite well depicted in this very nice movie, Indiana Jones and the Last Crusades, where he's hunting the Fountain of Youth, or actually he's trying to locate his father, who's hunting the Fountain of Youth. And in doing this, he has to, among others, combat the Nazis and many different booby traps in order to get to the Fountain of Youth. So this is something that humankind, I think, has been interested in for many years, um, and I think the focus in the scientific community today is not so much increasing lifespan, but more increasing a healthy lifespan. So for those of you that haven't seen this very excellent movie with Indiana Jones, you might be wondering if he actually found the Fountain of Youth and drank of it. And I can reveal that at least if he did, it didn't seem to work. So today Harrison Ford carries all the phenotypes of aging, including slightly more body mass and gray hair. So at least it didn't seem to work for him. So just before I get into the science, I just want to let you know a little about my background. So I'm currently an assistant professor at Department of Biomedicine at Aarhus University and the Stenus Diabetes Center in Aarhus. I have a background in human physiology from Department of Sports Science here in Aarhus. And after completing my PhD, I went on to do a postdoc with Professor Nels Jessen at the Department of Clinical Medicine, looking into fatty degeneration of skeletal muscle in type 2 diabetes. And then went on to a second postdoc at Stanford University with Tom Randall's group, where we looked at modifying muscle stem cell activity in order to enhance muscle function. So this was just a little bit about my background. So my agenda for you here today is, first of all, just to briefly explain a little on aging and skeletal muscle repair and maintenance. Then we're going to touch upon some different strategies to target and maybe rejuvenate muscle and muscle stem cells. And then finally, I'm going to show you some data on how we think the muscle niche is also modified under various circumstances and what we think might be driving this. So aging and healthy aging is something that's in particular focused this coming decade. So the World Health Organization has declared this the decade of healthy aging. For many different reasons, I think, but one of the obvious ones being that at this point there's over 1 billion people over the age of 60 in low and middle income countries. So this is a population that's quite rapidly expanding, and the ability to increase a healthy lifespan is, of course, something that is of interest to many people. So what are the hallmarks of aging? Well, Of course, there are many different traits of aging, and we still don't know exactly what causes aging in the first place. But one of the hallmarks is a decline in the functionality of stem cells, pretty much throughout the body. And this is probably driven by many different factors. One of them, the neighbor sitting right next to it, altered cellular communication. So, but this is going to be the focus of my talk for you here today. So for those of you that don't know skeletal muscle very well, um, skeletal muscle has an amazing ability to regenerate. So if you injure the muscle using a toxin, you can pretty much dissolve the entire muscle, and within a few weeks, you'll have more or less complete restoration of the muscle, new fiber formation, maturation, and eventually a fully functional muscle. And you can do this repeatedly, and the muscle will keep regenerating if it's a young, healthy animal. And the reason for this is, for a large part, the muscle stem cells. So muscle stem cells, as you can see here on this electron microscopy image from 1961, this is the first identification of these cells in skeletal muscle, done by Alexander Moreau, where you see these tiny cells sitting on top of myofibers. And by all means, they're pretty much inactive, doing nothing. And you can hopefully appreciate that the cytoplasm is also really tiny compared to the nuclei. So when the muscle is injured, these cells quite rapidly activate and proliferate and expand. And at some point, they start differentiating and fuse and form new myofibers. So we have a new fiber formation. And a small portion of these cells then return back to quiescence to replenish the stem cell pool to make sure that we can do this again and again. On the right here in pink, you have a muscle stem cell sitting on a human muscle fiber. So when we age, you have the age muscle down in the bottom panel here. When we age, the ability to regenerate is quite markedly impaired. So a few days after the injury, you still have tiny muscle fibers, and you have massive infiltration of fibrosis and sometimes also adipose tissue, resulting in eventually an impaired functionality of the muscle. And this becomes even worse if you re-injure the muscle. Whereas in young muscle, as I just told you, you have a complete restoration of muscle function. So in order to understand what drives this, Tom Rando and his group in 2005 published a paper in which they set out to investigate how the circulatory environment influences the functionality of muscle stem cells and the ability of the muscle to regenerate. So they did this by a technique called parabiosis, where they surgically joined, in this case, 2 young mice. And after a couple of weeks after surgery, these mice have developed a shared vascular system. So anything entering the circulation of one mouse will be delivered to the circulation of the other mouse, and vice versa. So after doing this, they then injured one of the muscles in one of the young mice. And as you can see here, you have new fiber formation, as it should be. Then they also stitched together 2 old mice and waited for a couple of weeks, and then did an injury in one of the mice. And as I just told you, regeneration was inefficient, and there was less new fiber formation and a massive fatty infiltration. However, when they then joined together a young mouse with an old mouse and injured the old mouse, what they observed was quite strikingly that the regenerative capacity of the old mouse was almost restored back to youthful level by virtue of sharing the circulatory system with a young mouse, highlighting that the environment in which these stem cells reside is really important in order for them to function optimally. So you could say that having a young partner might be beneficial, and it probably is, although sitting this close together for a human couple probably won't do anybody any good. So I'm not sure this is going to be a feasible strategy for humans. So the question is, how can we then rejuvenate the old environment? Well, one example of this is exercise. So in a recently published paper, Brett et al. Set out to examine if exercise could affect the ability of the muscle to regenerate. So they put mice in wheel cages and either allowed the mice to do voluntary wheel running, or they locked the wheels so that they couldn't actually run. So they did this for 3 weeks and then injured the muscle and looked at regeneration. So in the young muscle, they didn't really see an effect of exercise, consistent with the fact that regeneration seems to work fine in young animals. However, in the old animals, exercise seemed to remarkably almost restore the regenerative capacity of the muscle. So given that we know that muscle stem cells are really important for muscle regeneration, they of course looked into the functionality of these muscle stem cells. And what they found was actually that the ability of the muscle stem cells to activate when they extract them from the muscle is actually markedly enhanced in the old exercised animals. So exercise seems to increase the ability of the stem cells to activate, which is a really important feature of being able to regenerate your muscles. So of course they looked into what could be causing this and did an RNA-seq to screen for potential targets that would differentially regulate with exercise. And what they found was that the cycling D1 was quite markedly enhanced in the animals exposed to exercise, in muscle stem cells from the animals exposed to exercise. So cycling D1 is known to be key for the cells to enter G1. So they then went on to conditionally delete cycling D1 in muscle stem cells, and then looked at the ability of these animals to respond to exercise. And as you can see on the right, the rejuvenation of exercise was completely abolished in these animals, showing that cycling D1 was necessary for exercise to rejuvenate the muscle stem cells. So when you exercise or do physical activity, you of course also use the muscles. So this could be caused both by something in the circulation or alternatively something intrinsic in the muscle. And this is of course really difficult to answer, which is which. But what they did do was to collect serum from either exercising or non-exercising or exercising donors, and then they injected this serum into non-exercising recipients. And this actually phenocopied the effect that they found with just exercise. So just injecting the serum from exercising animals also enhanced the ability of the muscle stem cells to enter the cell cycle. So at least part of this seems to be mediated by something that's in the circulatory system. So to sum up this part of the talk, stem cell function is declining with the aging, at least in animals and in many different diseases. And this challenges the ability of organs to maintain and repair. However, these stem cells can be rejuvenated, and one of the means to do this is physical activity. So you might actually say that maybe Indiana Jones actually did become rejuvenated from all the exercise he did from trying to escape the various booby traps. I guess it just wasn't enough. So the systemic environment can rejuvenate or impair muscle stem cells. So another strategy would be to directly target the muscle stem cells. And we actually recently published one example of this. So in this paper by Antoine Desmarais and others, we found that the activity of muscle stem cells is actually quite different between different muscles. So in lower limb muscles, the activity of the muscle stem cells is actually quite remarkably lower than, for instance, in the diaphragm, here measured by EDU incorporation in vivo in the animals. And this increased activation of the muscle stem cells we found was correlated with a myogenic transcription factor known as PAX3, which we knew from previous studies was important for myogenesis during development. So using a conditional knockout model, we removed PAX3 specifically from the muscle stem cells. And when we did this, we more or less abolished the ability of the stem cells to activate, and surely also abolished the difference between limb and diaphragm that we see in wild type animals. This was completely gone in knockout animals. So why this difference between limb and diaphragm? Well, we knew from earlier studies that the length of the PAX3 mRNA transcript is different between muscle stem cells from hind limb muscles versus muscle stem cells from the diaphragm. And this difference in transcript length actually makes the mRNA degradable from being targeted by a microRNA. So when the transcript, the 3' UTR of PAX3 is longer, it can be targeted by a microRNA 206, which is not the case with a shorter 3' UTR. And we found that this small nucleolar RNA U1 has binding motifs, conserved binding motifs, just upstream of the polyadenylation site, the proximal polyadenylation site. So when U1 binds to these motifs, it will enable selection for more distal polyadenylation sites, resulting in a longer transcript that can be degraded, and you lose protein expression. However, in the diaphragm, you have less expression of U1, and thereby less binding of U1 to the motifs, resulting in more proximal polyadenylation sites, and thereby a shorter mRNA that can't be targeted by the microRNA. So by using this knowledge, we designed antisense in vivo morpholinos, which we injected into animals to block the U1 site specifically in the PAX3 transcript. And when we did this, we could actually increase PAX3 expression in the muscle stem cells, and thereby also increase the activity of the muscle stem cells. And so you might ask if this is something, this activity of the muscle stem cells, if this is something that matters. So, so when we conditionally deleted PAX3 in the muscle stem cells and waited for a while, we actually saw a decline in the size of the muscle fibers of the diaphragm, indicating that loss of this activity actually decreases the size of the muscle fibers. So we think this is something that's important for maintaining muscle size and a potential strategy to rejuvenate muscle stem cells. So of course, as a human physiologist, I'm interested in figuring out if this is something that's relevant in humans. So we recently sorted out muscle stem cells from human skeletal muscle, And indeed, we do see PAX3 expression in these freshly sorted muscle stem cells. And when we modify the length of PAX3 and thereby modify PAX3 expression, either increasing protein expression of PAX3 or decreasing, we can actually alter the activity of these freshly sorted muscle stem cells. So this mechanism seems to be conserved to human muscle stem cells and might enable us to actually directly target muscle stem cells through this mechanism. So now I'm gonna. So PAX3 is one example of a direct targeting strategy where you directly target specifically the muscle stem cells, for instance, using antisense morpholinos. So now I'm gonna switch gears a little, and instead of focusing on the muscle stem cells, I'm gonna focus on the muscle niche, which is also really important for maintaining muscle function. So as I already showed you, when regeneration is impaired, this is usually accompanied by infiltration of fibrotic and adipose tissue, which also impairs muscle function. This is something that we also see in many different disease conditions in humans, where you have this massive infiltration of fat and fibrosis, for instance, in this diabetic polyneuropathy patient compared to healthy controls. So this is something we see in many disease conditions and also in many different organs. So in mouse skeletal muscle, we've known for a while that the source of this fatty infiltration are these FAPs, or fibroadipogenic progenitors. So these cells reside in the interstitial space of skeletal muscle, usually quite close to the capillaries, and are mediating this fatty infiltration. So we were interested in trying to figure out which are the cells in human skeletal muscle that does this in order to be able to target these cells. So we also did single-cell RNA-seq, and we found this population, this quite robust population of FAPs in human skeletal muscle. And when we sort out these cells, they can quite massively form either adipocytes or fibroblasts, depending on the condition in which they're in. So these seem to be the source of fatty infiltration in human skeletal muscle. We became interested in this because we observed from previous studies that when we did RNA-seq on whole human skeletal muscle from type 2 diabetic patients, the pathways that really markedly popped up, either as biological processes or enriched pathways, were related to remodeling of the muscle niche or the extracellular matrix. So remodeling of the matrix is something that's really triggered in this disease condition, and probably markedly alters the functionality of the muscle. And when we looked histologically, we also found increased collagen expression and pyroleptin positive adipocytes in the muscles from type 2 diabetics. So in our study of these fibroadipogenic progenitors, we found a subpopulation of FAPs which express CD90 or Taiwan, which you can see expression of here in this UMAP plot. And also in this flow plot here, you see a population of CD90 positive FAPs. And what's interesting about these FAPs is that they have a quite markedly enhanced ability to enter the cell cycle compared to the ones that don't express CD90. So these cells can rapidly expand. And furthermore, when we look at collagen expression from these freshly sorted cells, they also have a much higher collagen expression. So combining the enhanced proliferation with increased collagen expression makes them really a key mediator, we think, of extracellular matrix accumulation in human skeletal muscle. So of course we looked for these cells in type 2 diabetic patients, and what we found was actually that they are increased in numbers in the type 2 diabetic muscles. And this was the only population we really found to be increased in the muscle from type 2 diabetic patients. So we have this model where we think that the degeneration that occurs with diabetes and severe diabetes of skeletal muscle is markedly associated with this specific subpopulation of FAPs, which have an enhanced potential to divide and also produce extracellular matrix, and thereby causing this degeneration of the skeletal muscle in these patients. So degenerative remodeling in type 2 diabetes, but probably also many other age-related diseases, is associated with an expansion of a subtype of FAPs in humans, and we think this is really causing this degenerative environment, impairing essentially muscle function. So this was a couple of examples of either stem cell related therapies or rejuvenation strategies to alter the functionality of these stem and progenitor cells in human skeletal muscle. And of course, there are many other potential strategies to do this. So I've just handpicked a few to show you here today. So if we're successful in rejuvenating humankind, we're not only saving ourselves, but maybe also saving the universe if we can rejuvenate Harrison Ford back to useful levels. So with this, I just want to thank you for your attention and thank my collaborators and mentors for support, and of course also financial support. I have made some questions to guide our discussion, which I really look forward to. I believe you will also receive these questions. So with this will be the end of my webcast. I look forward to discussing this with you soon. So now I will, terminate my talk, and if I can, there we go. So with this, I just want to say thank you for your attention, and we'll be in touch soon. Bye.